Dear Kuldeep, thank you very much for this kind introduction. Dear ladies and gentlemen, good morning. Dear Rajiv, thank you very much for the invitation. I'm very pleased to be here with you today, and I'm very excited and also pleased to see what's ongoing here at ICRISAT, especially under your supervision in the gene bank. You have the whole value chain in place actually. From the gene bank, we are pre-breeding to the breeders, to the nurse partners, and even to the farmers. This is great, and I would like to congratulate you. Today I would like to talk about crop wild relatives here, and I would like to also introduce you to a major initiative. This you have seen, all of you. We have many challenges ahead of us. We have population growth, climate change, limited resources, and the time is there already to utilize plant genetic resources for most crops in many regions. And you know this slide, or at least this study by Tanksley and Mankuch. And the idea is to broaden this elite gene pool by introducing beneficial genetic diversity from the crop wild relatives directly into the modern varieties. And this can be achieved today. But let me start at the beginning talking about the value of crop wild relatives also. So far, the greatest economic impacts in crop improvement have been in increasing disease and pest resistances. But we also have seen high levels of resistance or tolerance to abiotic stresses here. And you see a few examples here. Salinity tolerance has been improved by Helianthus paradoxus, or drought tolerance in breadwheat has been improved by introgression fragments from Aegilops deltoides. But there are so many obstacles for breeders in using crop wild relatives. And this is just very few considerations here. There's a whole bunch there. Of course, you know, these crop wild relatives, they show poor agronomic performance. And you can't predict a crop wild relative phenotype under field conditions. Or you can't predict a crop wild relative hybrid phenotype. Backcrossing might be needed, and this is time-consuming and challenging and expensive and difficult for various reasons. And also breeders, and we all have challenges around the understanding of excess and benefit-sharing provisions of germplasm. And then sometimes breeders don't know where to find these beneficial crop wild relatives. So, therefore, we have to consider and to discuss also the rationale here about plant genetic resources for breeding. And the idea is not to bring in just genetic diversity or neutral diversity. No, we need beneficial diversity for specific traits which are missing in the breeding material, which are not present in the elite gene pool. Therefore, for instance, if diversity for heat tolerance is already in the domesticated elite pool, there's no need to touch the crop wild relatives. But maybe, of course, there are other traits where diversity is missing. And then we encourage you to utilize and touch the crop wild relatives. And this is also an experience that Dr. Hari Upadhyaya made here in the Institute. And several of you might have seen this slide already. And the idea is, we have to discuss, and also Andreas Graner started here already, and I can build on this. Sometimes it's really needed to touch these crop wild relatives, and we really have to link the gene bank collections with the breeders. And this is challenging. And first of all, this sounds naive, but it's really important to define your target traits first. And then the plant genetic resource materials, they have to be prepared. Preparing means, for instance, single seed descending your material. You can phenotype the first life history traits, and then you can, will get a better overview about your material. But how to select your most promising materials? One idea is to establish so-called core reference sets or mini core collections. And I think for the first time, at least to my knowledge, all co-authors are here in this room. And the idea of core reference sets go back to Jean-Christophe and mini core to Harry here. And these core reference sets, the idea is to integrate data and to make these data available for all stakeholders. And you can build these core reference sets based on phenotypic and genotypic data, and also, of course, on ecogeographical information. And here you see it's really time-consuming, and you need a lot of labor to prepare these materials. And this is a picture from IPK where we single seed descended wild barley and wild wheat accessions. And at the end, this material will be ready to be genotyped or resequenced. And then you can select most promising materials and can feed them into your pre-breeding pipeline by crossing with elite materials. But even this is just the starting point. You need really long-term funding, and this is challenging. And your pre-breeding pipeline must be relevant for the industry. And then, and this is all pre-competitive research. And now let me please introduce a major crop wild relative initiative where we are trying to support for several years our partners. This initiative is supported by the Norwegian government. We have $50 million available for 10 years. We started in 2011. And it is a partnership with the Millennium Seed Bank. We have a strong capacity building component here, and we are focusing on developing countries. And we have 4 major parts in this initiative here. In the first part, we try to understand the natural distribution range of these crop wild relative species. We then prioritize the species and the gene pools. And then we started so-called gap filling by negotiating with partners. We have 24 collecting agreements in place now. And these crop wild relatives will be collected and conserved at several places. The next step is then pre-breeding. We have 19 pre-breeding projects, and I will come to this in a minute. And then information management is key here. And you see here, this is really a global initiative. We have more than 20 lead partners and more than 50 additional partner institutions in developing countries, even including NGOs and companies here. And here you see the pre-breeding project crops, starting from a forage crop here, alfalfa. We have chickpea here, and we have finger millet, which is important also for ICRISAT here, pearl millet, and where is pigeon pea here, for instance. And the idea was to work with partners in developing countries and to involve, whenever possible, also CG centers. This is a kind of overview about the current status. And you see in red, we had 2 pilot projects on rice and sunflower. And in green, these are the pre-breeding partnerships. Some of them finished already. But we invited from these 19, 10 to 12 project partnerships will be invited for next phase, for an evaluation phase. And you see most projects, they are running 3 to 5 years, and we are trying to support even longer. Here you see the pre-breeding partnerships important for ICRISAT. We have 3 of these. Finger millet, this is with ICRISAT in Kenya. Pearl millet and pigeon pea, we have partners here in India, and we have ICRISAT and Jonas partners here for pearl millet. Several companies are involved, and 2 days ago we saw, for instance, the research station in Warangal. I was really impressed to see this material. They are promising and breeders are involved, defining the traits of interest. Really great. And you see on the right-hand side our focus traits, mainly abiotic stress, but for some crops we need also biotic stress-related traits. Here, this is our key partner here at ICRISAT, Dr. Shivali Sharma, but she gets great support from her breeders. There is Dr. Kumar, for instance, Samir Kumar, or Dr. Gupta, and a whole bunch of other people behind her. Really great to work with powerful women here. But the original ideas here, they go back also to Dr. Hari Upadhyaya. And then Rajiv decided with Hari to hand over to young women. This is really great. Just a few words about these partnerships here at ICRISAT. First one is on pigeon pea. Here we can build on material they were developed already back in 2010, right, or 2012. And so this is quite advanced material. And so we really can see some first significant progress here. And I learned that ICRISAT and partners, they found the first multiple resistant high-yielding line. And this one is resistant to wilt and sterility mosaic disease and even Phytophthora. And in these material, they go into national programs and might be released in the near future. But there are more other high-yielding lines in different duration groups. Here for pigeon pea, we have different duration groups, early and medium, and so on. But also these materials are being tested for their maintainer and restorability, and might be beneficial for hybrid breeding in the near future. This is the second one. This is on pearl millet, and here we really initiated the first crosses here. And the idea is to develop and to advance 4 backcross populations. And we've discussed this yesterday. The material has been in the field this year. It was quite challenging. The weather conditions were challenging. And more advanced material will be in the field also next year. This is quite early. This is BC1 F2 material and sometimes further developed. Here we need more time. And the third project here is on finger millet. You see Damaris here. She couldn't make it. She had just a striker workshop in Kenya. So I'm sending her your best regards. And here she has several objectives. She is screening wild but also breeding lines and farmer preferred varieties for resistance to striker. And this striker is really an issue in Eastern Africa. She's developing also interspecific populations, and she's also developing molecular markers for marker-assisted selection. This is just one picture here showing the difference between infected and non-infected plants here. And these infected plants, they are of course shorter, and they can show necrosis and wilting. But there is hope that we might change this in the future, and because here you see 6 days after an infection, the number of first leaf primordia unattached striker. And you see here, for instance, the susceptible cultivated line, the tolerant cultivated one, and here's a tolerant wild one. So significantly less primordia at this stage, 6 days after infection. Let me talk briefly about the outputs of this Crop Wild Relative Initiative. Of course, we are producing Crop Wild Relative seeds. Not producing, we collected these and make them available under standard material transfer agreements. They will be stored at gene banks at the national and international level, and finally also be backed up at Svalbard. Then, most important pre-bred lines will be made available freely under SMTA. Then capacity building is very strong here. We are running workshops and other initiatives. I would like to focus on the data now, and we have several possibilities to make data available. We have our project webpage, we have Genesis and Germinate 3. This is our project webpage here. Sorry. For instance, we have a kind of crop wild relative inventory where we are documenting more than 4,000 use cases for crop wild relatives. This has been recently published in Crop Science. Then we have an occurrence database, an atlas, for instance. We have Genesis. Genesis is a kind of online portal to these plant genetic resources. You can order seeds via Genesis, and later on you will be able to link genotypic and phenotypic data. Every accession will get a DOI. And we have pre-breeding data storage solutions. And let me briefly talk about Germinate. We have, where's David Marshall? He was here before. And this is a kind of generic plant resource database developed at the James Hutton Institute in Scotland. And I got these slides 2 days ago from Paul Shaw and Sebastian Raubach. You know, there are several challenges ahead of us. We have to store and manage these data and make them available to all stakeholders freely and online. And Germinate 3 is a single central repository, and this Germinate 3 solution can handle these data and can also visualize these data sets that pre-breeders produced. This is an example here from our eggplant pre-breeding partnerships. At the moment, we have information for 500 accessions available. We have molecular markers included, and many other informations are available. And this can be customized, but we were using a common interface here. All kind of accession information can be stored here and analyzed. Collection sites, for instance, even metadata are accessible, a great tool. Trait data can be visualized here. You can select the traits, you can plot these, you can make marker and map information available, all the data sets that you would like to share with the community. This is an older example here from rice pre-breeding with Cornell and Erie. And in red, you see the elite background, and in green, you see the crop wild relative introgressions visualized. We have links from Germinate 3 to Flapjack in this case, or even other tools can be integrated. For instance, Helium. This is a new tool to visualize your pedigree data. And on top of this, you will have genotypic data and phenotypic data. And currently, the James Hutton Institute is working hard to build several instances for this crop wild relative project. And you see the different crops here, which are important for different continents. Let me briefly summarize here. So crop wild relatives really, they are large reservoirs of beneficial diversity that we will need certainly in the future for crop improvement. And as IPK already started and you here also at ICRISAT, we have to close this gene bank breeding divide. By making materials available, we have to characterize these materials. And pre-breeding needs long-term funding support. Public-private partnerships, as we also discussed yesterday, are very important here. And we are at the Crop Trust, we are convinced that Germinate 3 is a great tool to visualize these data that we are producing here with the partnerships, and that we can effectively share these data sets. And this will also help the breeders later on to make the right selections for their breeding pipelines. I would like to acknowledge all partners from our initiative here, and also the James Hutton Institute for developing the Germinate 3 instances for us. Thank you very much for your attention. Thank you.
Thank you, Benjamin. You saved a lot of time. I think that would be good for discussions. And we have sufficient time. Any questions for Benjamin? Yes.
Benjamin, you pointed out several areas where you evaluate wild germplasm in terms of the usability for crop plant improvement. But one major bottleneck that is sort of shunned by plant breeders is the limited recombination between crop wild relatives, also in the primary gene pool and elite germplasm. Is there any activity within the program you just described to address this issue?
Every partnership is starting from different, let's say, starting points, of course. Sometimes we have to do the first crosses. Sometimes the species boundaries are not clear. So they are just trying to make these crosses using tools. We have to go for embryo rescue, but we are not specifically addressing in a scientific way this recombination challenges. Of course, at the end, we have in nearly every, I think in every project, we have also kind of genomics part or molecular markers part included. And these genomic markers might at least help us to select the right materials by using Flapjack, for instance. But there's no research activity ongoing here. The focus is on pre-breeding and involving farmers.
We have first Jonathan first there. Yeah. Please.
Hi, thanks very much. So one challenge in making the results of such pre-breeding available is the Nagoya Protocol. I just wondered how you manage that. Let's say if you access germplasm, some segments are bred in, and then some of your partners like Pioneer might want to turn it into a variety. So how is that a problem? And if so, how do you deal with it?
Yeah. First of all, as I mentioned, we are ready to share under SMTA conditions. So everybody that is signing this SMTA of the plant treaty will receive the seeds. And this is pre-competitive research. It's so maybe in the future we have to discuss these this issue again. Yeah. So far, if a breeder signs the SMTA, he will get the material.
Cheralu from PJTSAU, India. Many of the leading centers in major and as well as the minor crops throughout the world actually forgotten the pre-breeding. Everybody is interested in releasing the varieties having the high yield or something like that. But there is no material from many of the centers which are really troublesome in increasing the yield potential of the crops, both for the yield and also biotic and abiotic stresses. Why can't the project like this identify some of the major yield limiting factors in different crops and take lead in improving or trying for the pre-breeding efforts and distribute the same thing to the private and public sector? So that this may be one of the major way through which we can get a faster improvement in different crops. Instead of going for the generalized improvement and creating a lot of variability, so many things. This is my personal view. Thank you.
To that? The question is primarily related that most of these crops would not have the good pre-breeding programs, especially for increasing the yield or the productivity. He's saying, can we have some of the projects where productivity could be a primary trait for pre-breeding? So, minor crops specifically for the minor crops. pre-breeding for enhancing productivity?
Well, I think the question is there is more effort need to be in the pre-breeding area, and emphasis needs to be given on yield improvement. I think that's what he has been showing. So I think that's a comment. We take that comment. Move to Hari, please.
I'm Hari Upadhyay, and my comment is respond to Andreas. Yes, when you go to tertiary or secondary gene pool, obviously there is a less of recombination. And at ICRISAT, since 1972 it was established under wide hybridization program. This work was initiated in pigeon pea as well as in groundnut. And in pigeon pea, a variety was really released, a line ICPL 87162 with high protein content by hybridization with Cagenes scaraboides. Later on, using the tertiary gene pool and secondary gene pool, including platycarpus, there are lines, introgression lines available which have some promise even for helicoverpa. I say some promise, still it has a lot of drags and loads of the undesirable genes, so that needs to be cleaned up. So these efforts are not that they are not there. Only thing is that very successful have come to the public domain. Efforts are going on here as well as at several places.
Baba, you have a question?
Thank you very much. Chikemba from the Food and Agriculture Organization of the UN. Congratulations, Benjamin, on this presentation. By definition, the products of pre-breeding should be advanced materials that plant breeders in the public or private sector can use to develop varieties. Yet I see in your presentation that you're actually distributing these pre-bred materials directly to farmers. I would want you to clarify regarding, the stability of these materials, whether they really qualify as varieties, and also how you take care of the quality assurance for the seeds. Thank you.
Thank you very much for this question. Of course, it's a very kind of complex question. The idea is to prepare these pre-breeding, to develop and prepare these pre-breeding material, and to develop them further in this partnership. Only at the very end, farmers will get involved. When the material is stable, and there are different streams in this project. I had no time to discuss this. There's one stream we call pre-breeding. We're involving pre-breeders, breeders, and then there's the other stream where we will try to go for at least participatory varietal selection. And we also understand that participatory plant breeding is challenging. And in most countries, we have this pipeline here in place, for instance, at ICRISAT. We need several years of pre-breeding, and then our DNAS partners here will take care to release these material. Farmers will be involved in defining the traits of interest and to maybe select together with the breeders. But at the very end, farmers will also get these released varieties. At the very end. And of course, we are trying to be efficient here, but also we have a kind of mission to improve the capacities in developing countries. So for some crops in some countries, we also will try participatory plant breeding. For instance, in sorghum in Western Africa. Also maybe the impact might be different. I hope I answered this.
The last question from here now.
Yeah, Benjamin, my question is very simple, not complex. So you are selecting, my feeling is that selected crops under this initiative.
Yes.
Is groundnut in that list?
No, Unfortunately, not yet.
I saw yesterday evening you were enjoying beer with peanuts, right? And if I saw like maybe the cultivated, the primary gene pool is, you can say, so much isolated that it is required that such an initiative so that the diversity that is within the wild accessions can be harnessed. Yeah. So I need that should be the number one or maybe top 5 crops in priority.
Okay, we can discuss that on a beer and peanut this afternoon.
Sure.